Imagine a creature that looks like it swam straight out of a forgotten Saturday morning cartoon. Seriously. Like a truly bizarre, googly-eyed mixture of a tadpole, maybe a horseshoe crab, and, yeah, even Gary the Snail from SpongeBob. Sounds like something from a fever dream, doesn't it? But this isn't just some quirky ancient fish. It's actually a profound discovery that's currently upending everything we thought we knew about early animal evolution. See, for as long as many of us can remember, the story of life's transition on Earth, well, it's been told with a pretty widely accepted timeline. It began with simple, often sort of sedentary bottom feeders. Then it progressed to complex jawed predators that dominated the ancient oceans. And eventually that bold leap to land-dwelling creatures culminating in, well, us and everything else we see today. But today we're diving deep into a story where a tiny fossil fragment, barely the size of your fingernail, is forcing scientists to completely rewrite one of the earliest chapters in animal evolution. It's a story with unexpected twists and turns, far from a straightforward march of progress.
And that's precisely the mission of this deep dive.
Yeah.
We're going to explore how an international research team, armed with some really cutting-edge technology, was able to peer inside this 400 million-year-old mystery. And they revealed anatomical features previously believed to be exclusive to much later, far more evolved creatures. This isn't just about understanding one peculiar fish. It's about understanding the complex, often nonlinear, and frankly surprising paths evolution takes. It's also, I think, a testament to how new tools and technologies can illuminate those large data gaps that have long existed in the fossil record. They push the boundaries of what we thought was knowable about ancient life, and it demonstrates the dynamic nature of science itself, really.
Indeed. Today we're not just looking at old bones or, dusty archives. We're unpacking a truly fascinating story of persistence, technological innovation, and scientific discovery. It's a narrative that fundamentally reshapes our view of the past, drawing direct connections between the earliest forms of ocean life and, well, the very origins of our own vertebrate body plan. So get ready for some truly jaw-dropping revelations. Okay, part one, the Norsel-Aspis revelation, a shark heart in a lamprey skin. To start, let's delve into the star of our deep dive, the creature forcing us to rethink so much, Norsel-Aspis glacialis. Before we even get into its astonishing internal anatomy, tell us about its rather unusual and, frankly, protracted discovery. It sounds like a real tale of paleontological patience.
Oh, it truly is. It's a remarkable story, one that spans decades and really highlights the immense dedication often required in paleontology. Sometimes, discoveries are made years, even decades, before their true significance can be understood. The fossil itself was actually unearthed way back in 1969. Can you imagine that? Over half a century ago. It happened during a French paleontological expedition to the Arctic Spitsbergen archipelago in Norway, a really remote place. The team collected thousands upon thousands of sandstone blocks, each potentially containing fossilized remnants of ancient life. It's like searching for a needle in a haystack, isn't it? Except the haystack is made of rock and, well, extends for miles.
Wow, thousands of blocks.
Exactly. But this particular fingernail-sized fossil fragment, described as a perfectly preserved, half-inch long Norsel-Aspis cranium, well, it wasn't immediately identified or studied in detail. It remained hidden among those thousands of blocks, waiting patiently in an archive for another 4 decades. 4 decades, before researchers could even dedicate enough time to systematically sort through them all. It was only then, after decades of patient curation and methodical sorting, that the study co-authors, Philippe Janvier and Pierre Guerriot, split open the specific rock that held this pivotal snapshot. A snapshot from the Devonian period, dating back an astonishing 410 to 407 million years ago.
40 years waiting in a drawer, essentially.
Pretty much. So while the initial collection was relatively quick, the actual revelation was a slow burn. It's a testament to the sheer volume of material paleontologists often work with and the immense time it takes to process it all. Sometimes it feels like these fossils are truly just waiting for us to catch up with the technology they need to reveal their secret.
That's an incredible timeline for a discovery. It really makes me wonder, how in the world did they get so much detailed information from such a tiny ancient fragment, especially considering it was encased in rock? What makes the approach they use so revolutionary compared to, say, traditional paleontological methods? And what kind of details could they even hope to extract from something so old and small?
Right. This is where the story truly shifts gears and highlights a real paradigm change in the field of paleontology. For a very long time, traditional methods often involved physically grinding away extremely thin layers of a fossil, bit by painstaking bit. to expose internal structures. Imagine taking a geological slice, meticulously drawing what you see, then grinding off a microscopic layer, and repeating that process thousands of times, almost like creating a flip book of internal anatomy.
Okay, I can picture that. Labor-intensive.
Incredibly so. And while pioneering paleontologists like Eric Stensio used these serial grinding sections to provide foundational knowledge about early vertebrates, it was an inherently destructive process. And crucially, it was also highly prone to misinterpretation. Tiny bits of damage or taphonomic artifacts.
Taphonomic. What's that?
Good question. Taphonomy is basically everything that happens to an organism after it dies until it's discovered as a fossil. So taphonomic artifacts are things like cracks or mineral infills caused by the fossilization process itself, not part of the original animal. And with grinding, these could easily be mistaken for actual anatomical features, which could lead to a skewed understanding of the organism's true biology.
I see. So you could easily misread the evidence.
Precisely. But with Norsel-Aspis, the team turned to a truly cutting-edge, non-destructive technique, synchrotron-based X-ray microtomography, or PPCS-CT for short. They sent the specimen to Switzerland's Paul Scherrer Institute, which houses a powerful particle accelerator, a synchrotron. Now, what makes this revolutionary is that instead of grinding, this technique uses incredibly high-energy X-ray beams. But it doesn't just measure how much X-rays are absorbed, like a medical X-ray. It also measures the phase shift of the X-ray beam as it passes through the specimen.
Phase shift.
Yeah, it's a property of the wave. It allows for unparalleled contrast, which is absolutely crucial for fossils, because often you have very subtle density differences between the fossilized tissue and the surrounding rock matrix. The X-rays scan the specimen layer by layer, producing exquisitely preserved digital slices at submicrometer resolution. We're talking details as small as a millionth of a meter. Think of it like a medical CT scan, but on a vastly more detailed and sensitive scale. It lets scientists see through the rock without harming the precious fossil inside.
Wow. Okay, so non-destructive and super high res.
Exactly. And after thousands of hours of digital reconstruction, and I really mean thousands of hours of painstaking work by fully skilled technicians and paleontologists, these digital slices were stitched together to create a full 3D model, and the results were truly jaw-dropping. The 3D scans revealed these delicate films of bone that enclosed the fish's organs with astonishing detail. We're talking about capturing the ghosts of organs, not just bone, but the impressions left behind, its heart, its intricate network of blood vessels, the precise shape of its brain, the delicate pathways of its nerves, the structures of its inner ears, and even the tiny muscles that moved its eyeballs.
Even the eye muscles from 400 million years ago.
Yep. The resolution was phenomenal, down to 1/100th of a millimeter wide. This level of detail allowed researchers to differentiate between true anatomical structures and, fossil damage, or those taphonomic artifacts we talked about. Something nearly impossible with older techniques. For example, earlier interpretations, based on less precise methods, had suggested Norsel-Aspis had a single venous drainage system, like a primitive lamprey, or maybe an endoskeletal joint in its pectoral fin attachment. The new scans conclusively showed these were, in fact, an artifact of damage to the original specimen. Basically cracks or mineral intrusions mistakenly identified as anatomy.
So it corrected previous misinterpretations, too.
Fundamentally altered our understanding, yes. As the lead author, paleobiologist Tetsudo Miyashita, rightly said, with this exquisite digital atlas, they now know Norsel-Aspis in greater anatomical detail than many living fishes. It allowed for sweeping updates to its interpretation, revealing it to be surprisingly jawed vertebrate-like and anatomically not as primitive as previously thought. It's truly seeing ancient life with modern eyes.
Okay, so with these unprecedented details revealed, what did the scientists actually discover about Norsel-Aspis's internal biology? What was truly groundbreaking about its anatomy, especially for a jawless fish from that ancient time? I know the sources use this incredible phrase, the heart of a shark under the skin of a lamprey. Can you break that down for us and explain the paradox?
Yeah, that phrase, coined by Tetsudo Miyashita, it perfectly encapsulates the central paradox of Norsel-Aspis glacialis. Despite being unequivocally jawless, it shares that fundamental external characteristic of primitive fish like lampreys. Its internal anatomy was different. As revealed by the synchrotron scans, it possessed sophisticated features previously thought to exist only in much later, more active jawed species. It was an astonishing evolutionary mosaic, a blend of features that really challenges our expectations.
A mosaic. Okay, so different parts evolving at different rates.
Exactly. Let's start with its heart of a shark, its circulatory system. This ancient creature had an extremely powerful heart and widened vessels facilitating greater blood flow. This is a stark contrast to what we'd expect from a primitive, generally kind of sluggish, jawless fish.
Hmm.
Unlike the more primitive lampreys, which have a single, simpler venous drainage system, Norsel-Aspis had paired common cardinal veins, like in modern jawed vertebrates.
Paired veins. Why is that significant?
Well, this paired system is crucial because it drains a greater volume of blood into a large sinus atop the heart, and that directly supports higher metabolic rates and sustained activity. Imagine trying to run a marathon with a small, inefficient engine versus a powerful, multi-cylinder one, right? The difference in blood flow directly translates to how much energy an animal can generate and sustain. Furthermore, the presence of a cardiac branch of the vagus nerve suggests sophisticated cardiac control.
Meaning it could regulate its heartbeat.
Precisely. It means it could regulate its heart rate and blood flow with precision, something vital for dynamic movement, whether for pursuit or, as we'll see, evasion. This advanced circulatory capacity is exactly what Miyashita was referencing with that heart of a shark comparison. Sharks are known for their high metabolic rates and powerful, sustained swimming abilities, all powered by efficient, high-volume hearts. Scaled to human size, Norsel-Aspis's heart would have been, get this, as large as a cantaloupe melon.
A cantaloupe, wow.
Yeah, indicating immense horsepower for its activities, far beyond what its simple external appearance suggested.
Okay, so powerful engine. What about its senses? Were they equally advanced?
They were, far from primitive. For instance, consider its eyeballs. It had 7 tiny muscles to move its eyeballs, which is actually one more than humans possess.
No.
Yeah. And this is a fascinating detail because it suggests that 7 was likely the ancestral configuration for vertebrates, and that both lampreys and jawed vertebrates, like us, independently lost one muscle during their evolution. So Norsel-Aspis actually retained a more ancient yet complex configuration here. It gives us a glimpse into the very earliest eye movements. But the sophisticated design didn't stop there. Even more striking were its outsized inner ears.
Outsized how?
Proportionally huge. Scaled to human size, they'd be each the size of an avocado.
Avocado-sized ears. What were they for?
Well, these enlarged inner ears, with specialized internal structures like an enlarged pars inferior and a columnar sinus superior, they were absolutely crucial for sensing vibration, orientation, and acceleration. This enabled precise navigation in a complex, three-dimensional aquatic environment. It could detect subtle changes in water currents or the movement of other organisms. Such structures are actually considered derived features of jawed vertebrate inner ears, which just underscores how advanced its internal sensory capabilities were. And beyond its ears and eyes, Norsel-Aspis also possessed these complex sensory fields on their head shield.
Sensory fields, like patches on its head.
Sort of, yeah. These specialized fields have potentially detected waterborne vibrations or even generated mild electric fields for navigation, maybe in murky waters, a bit like how some modern fish sense their surroundings. So this whole suite of acute senses meant Norsel-Aspis was incredibly aware of its surroundings, a true master of its immediate environment.
So powerful heart, amazing senses. How did that translate into movement? You called it action-packed?
Right. This powerful internal machinery, working in concert with these acute senses, translated into remarkable agility. Its action-packed anatomy for locomotion. It sported these tilted, paddle-like fins behind the gills. And these weren't just for passive stability. Their unique angle and configuration suggest they were specifically adapted to allow it to quickly turn, stop, and speed up as needed. This made Norsel-Aspis something of a sports car among the generally sluggish, jawless fishes of its time. It could execute rapid changes in direction, bursts of speed, and generate both thrust and lift. Unparalleled maneuverability among its contemporaries.
It sounds like Norsel-Aspis was incredibly well equipped for its time, truly a sports car among its peers. But if it lacked jaws and teeth, what was all that speed and precision for in the Devonian oceans? What was its role in that evolving ecosystem?
Precisely. And this is a critical point that kind of flips our traditional understanding of why such sophisticated adaptations might evolve. Given Norsel-Aspis's fundamental lack of jaws and teeth, it was likely a bottom feeder, maybe relying on suction or filtering for nourishment. This sophisticated anatomy was likely designed for evasion, not hunting.
Defense, not offense.
Exactly. It was built for escaping. The Devonian seas were becoming increasingly competitive, a true evolutionary arms race. While Norsel-Aspis lacked chompers, other species were rapidly evolving them, becoming formidable predators. So Norsel-Aspis needed speed, agility, and keen senses just to survive the new predatory pressures. Think of it as a highly specialized defensive mechanism where defense rather than offense drove these early sophisticated adaptations.
Okay, so it developed these advanced features to avoid being eaten.
Right. But this action-packed defense mechanism, though, it turns out to be a key to understanding later offensive capabilities. This phenomenon is a classic instance of what we call pre-adaptation.
Pre-adaptation.
Yeah, think of it like a smartphone that was initially designed purely for communication, right? But then, through its robust processor and versatile camera, it suddenly becomes the perfect platform for sophisticated augmented reality apps, something completely unforeseen initially. Here, the very traits that developed under one selective pressure defense and evasion became instrumental for a new, fundamentally different function of fence and predation once JAWS finally evolved. It implies that the evolutionary trajectory towards becoming an apex predator wasn't a sudden singular leap once JAWS appeared. Instead, it was a complex, multi-stage process built upon a foundation of pre-existing sensory, circulatory, and locomotor enhancements. Enhancements that initially served entirely distinct survival needs, like escaping.
So the groundwork was already laid before JAWS even showed up.
Yeah, exactly. This is why Norsel-Aspis challenges a simplistic linear view of evolutionary progression, where all features evolve from simple to complex in lockstep. It was not simply a primitive fish, it was a sophisticated creature with a unique combination of traits. It exhibits what we call evolutionary mosaicism, where different organ systems or traits evolve at varying rates. This continuous refinement of anatomical data, especially with new technologies, demonstrates that the distinction between primitive and derived traits in the fossil record isn't static. It's a constantly evolving understanding.
That makes sense. Science is always updating.
Absolutely. And I should probably add a note here about how scientific understanding itself evolves, even after groundbreaking discoveries like this. While the Norsel-Aspis study provides these incredible revelations, there's always ongoing scientific debate and refinement, of course. Some critiques have been raised, specifically regarding its phylogenetic context, basically, where it fits on the evolutionary tree. It's been argued that taxon exclusion marred this otherwise excellent report, meaning maybe they didn't include all the relevant comparison groups.
Okay, so the interpretation depends on which relatives you compare it to.
Precisely. And some argue that osteostracans, the group Norsel-Aspis belongs to, may be basal only to sturgeons, rather than broadly representing the sister group to all jawed vertebrates. If this alternative phylogenetic placement is accurate, it would sort of narrow the broader evolutionary implications of Norsel-Aspis's gnathostom-like features. It would suggest these advanced traits might be specific adaptations within the lineage leading just to sturgeons, which themselves, interestingly, use extendable ventral tubes for feeding, not typical biting jaws. So it wouldn't necessarily represent the condition right before all jawed vertebrates evolved.
I see. So the exact placement on the family tree changes the story a bit.
It does. This ongoing discourse highlights a fundamental principle of scientific inquiry. The interpretation of evolutionary significance, even with revolutionary new data, is heavily dependent on the phylogenetic framework you use. Incomplete or biased phylogenetic sampling can lead to overgeneralizations or misinterpretations of evolutionary patterns. It underscores the iterative nature of scientific progress, where new data are constantly integrated into and sometimes challenge existing theoretical constructs. It's really a testament to the self-correcting nature of science.
Part 2. Rewriting a evolutionary script. Jaws, shoulders, and surprising paths. That's a fascinating reframe that evolution isn't always a straight line. For so long, the accepted theory of early vertebrate evolution was that jaws were the foundational evolutionary leap.
Right.
That single innovation, having a mouth that could bite and chew, was thought to drive all other subsequent adaptations for a predatory lifestyle, from stronger hearts to sharper senses. How does Norsel-Aspis fundamentally challenge that jaws first, then limbs kind of hypothesis?
Yeah, this really does turn the narrative on its head quite profoundly. The traditional view, as you said, saw jaws as the singularly pivotal event, the thing that then spurred the extraordinary variety and abundance of Devonian fishes. The idea was, once you could bite, everything else, your heart, your senses, your muscles, needed to catch up to support that new, active, predatory lifestyle. It was seen as this single, transformative event. But Norsel-Aspis provides compelling evidence that acute senses and a powerful heart evolved well before jaws and teeth were even a glimmer on the evolutionary horizon.
So the support systems came before the main event, so to speak.
Exactly. These sophisticated internal systems, with their capacity for higher metabolic rates and rapid responses, they were already in place. They were initially developed, as we discussed, for evasion, for outmaneuvering the nascent predators in those competitive Devonian seas. This means these early fish were, in essence, pre-adapted for a high-energy active lifestyle before the advent of jaws. So when jaws did finally evolve, they weren't building from scratch. They integrated into an already highly capable sensory and swimming system. Think of it like adding a powerful, finely tuned engine to an already very capable chassis. Allows for much greater speed and maneuverability, rather than building the entire sports car from scratch just when the engine appeared.
Okay, that makes sense. It leverages existing capabilities.
Right. This integration then, quite logically, led to the explosive diversification of Devonian fishes, because the fundamental physiological and sensory groundwork for active predation was already laid. It implies a much more gradual and incremental accumulation of advantageous features over time, rather than a single saltational, meaning sudden, large step leap, driven by one innovation like Jaws. So it wasn't a simple linear progression from a passive bottom feeder directly to an apex predator.
So it definitely wasn't a straight line from bottom feeder to apex predator. That's a significant shift in perspective, and quite counterintuitive to the simple story we've often been told.
No, absolutely not a straight line. As Miyashita noted, it wasn't as simple as marching straight from a bottom feeder to an apex predator. This discovery highlights the concept of evolutionary mosaicism, which is a recurring and critical theme in deep evolutionary history. It's where different traits or organ systems evolve at varying rates, creating organisms that are a blend of ancestral and derived features. Norsel-Aspis, with its jawless, lamprey-like external form combined with a shark-like heart and advanced senses, perfectly exemplifies this. It was not merely a primitive fish waiting for jaws to become complex, it was a highly sophisticated creature in its own right, uniquely adapted to its environment. This challenges any simplistic linear view of evolutionary progression where all features evolve from simple to complex in lockstep. It reveals a far more intricate and opportunistic evolutionary process.
Okay, beyond the story of Jaws, it seems Norsel-Aspis continues to hold more surprises for us, even shedding light on parts of our own anatomy. What other long-held evolutionary theories did this tiny fossil challenge, and how does it connect to something as fundamental as our shoulders and arms?
Yes, this is another truly significant revision to a long-held hypothesis about our own body plan, specifically concerning the evolution of the pectoral girdle.
The shoulder blades and collarbones, basically.
Exactly. The bony arch that supports the forelimbs and vertebrates. For a very long time, it has been theorized that the shoulders, and by extension the arms in modern tetrapods, that's us, four-limbed vertebrates evolved from modified gill structures. The idea was that some of the skeletal elements associated with the gill arches, those cartilaginous or bony supports for the gills in fish, were repurposed, transformed over millions of years to become the pectoral girdle and forelimbs. This was a classic example of evolutionary repurposing, a kind of biological recycling.
Making new things from old parts.
Right. But Norsel-Aspis presented clear counter-evidence. Researchers meticulously traced the nerve pathways within its fossilized cranium, a feat made possible by the incredible resolution of those synchrotron scans. And what they found was definitive. A nerve linked to Norsel-Aspis's shoulder specifically, the brachial plexus nerve, which innervates the pectoral fin, was entirely separate from the nerves reaching the gills.
Separate nerves, so they weren't connected in the way the old theory suggested.
Precisely. This is clear evidence that one did not come from the other. It directly contradicts the gill arch hypothesis for shoulder origin. It suggests a different independent evolutionary pathway for our shoulders. This anatomical disjunction, the fact that the nerve supply to the shoulder was distinct from that of the gills, means the shoulder couldn't simply be a modified gill arch structure.
Okay, so if not from gills, where did shoulders come from?
Well, the new theory, bolstered by Norsel-Aspis, posits that the shoulder evolved as a wholly new structure. It arose from a distinct developmental domain, not a repurposed gill arch, an innovation. This innovation then led to the formation of a separate neck region. See, in primitive jawless fishes, the head was continuous with the torso, essentially. No neck. Imagine a fish where its head just blends seamlessly into its body, but jawed vertebrates evolved a neck and throat, allowing for greater head mobility, which is crucial for a predatory lifestyle, right? Enabling them to quickly snap their jaws or scan their surroundings.
Makes sense. A neck gives you more flexibility.
Absolutely. And Norsel-Aspis, in its mosaic anatomy, kind of straddles both anatomies. Its head is directly attached to the shoulder without a true neck, almost as if our arms were sticking out behind their cheeks. But while it didn't have a distinct neck in the modern sense, its elaborate head-trunk interface, with its enhanced inner ears, sophisticated shoulders, and powerful heart, may have been a crucial precursor. This complex arrangement might have facilitated the later emergence of a muscular neck region between the pectoral girdle and the skull. And this new neck provided the increased head mobility and mechanical advantages that would become critical for the efficient function of jaws in later vertebrates, allowing for more agile hunting and manipulation of prey.
So our shoulders might be a completely new invention, not just recycled gills.
That's what this suggests. A novel evolutionary invention, decoupled from the gill system, which profoundly reshaped the vertebrate body plan, and it laid the groundwork for our own terrestrial locomotion eventually.
Part 3. A modern parallel, the pseudobranch in the ancestral jaw. That's incredible. A single ancient fossil rewriting so much of our understanding of early evolution. It really highlights how interconnected all these evolutionary paths are. But our deep dive doesn't stop there. This conversation around ancient fish and the origins of jaws actually has direct connections to complex, ongoing, modern-day biological debates. Let's shift gears and talk about something called the pseudobranch. What exactly is it, and why has it been a source of such contention in evolutionary biology, specifically regarding jaw evolution?
Ah, the pseudobranch. Yes, it's a fascinating and somewhat enigmatic structure found in many fish. It's essentially a gill-like epithelial elaboration that sits behind the jaw of most fishes. If you were to look inside the mouth of a typical fish, behind the upper jaw, you'd find this small, often reddish gill-like structure. For a very long time classically, it was regarded as a vestige of the ancestral gill arch-like condition of the nithostom jaw.
A vestige, like a leftover bit.
Exactly. The prevailing hypothesis for over a century was that our jaws, meaning the jaws of all jawed vertebrates, or nithostomes, evolved from an ancient gill arch. Specifically, the first gill arch was thought to have transformed into the jaw, and the pseudobranch was considered a reduced, non-respiratory remnant of that ancestral gill, a little echo of it. This idea of jaw evolution by the transformation of a gill arch has been a cornerstone of vertebrate comparative anatomy, rooted in observations of apparent correspondence between jaw segments and gill arch skeletons.
Okay, so the standard story was first gill arch becomes jaw, pseudobranch is a tiny leftover.
Right. However, more recently, this jaw evolution by transformation of a gill arch hypothesis has faced significant challenges. Some researchers, like, interestingly, Tetsuro Miyashita, again, yes, the same Miyashita from our Norsel-Aspis discussion, showing how interconnected these fields truly are, suggested an alternative. He proposed that the pseudobranch was not a vestigial first gill from the mandibular or jaw arch, but rather a specialized derivative of the second hyoid pharyngeal arch, the arch behind the jaw arch, and that it evolved instead for a non-respiratory, maybe chemosensory function, perhaps detecting changes in water chemistry as food passed by.
So a different origin and a different function. Why did they think that?
Well, the reasons for this challenge were primarily based on observations of the pseudobranch in adult fish. First, it receives oxygenated blood from the efferent hyoidian artery, which seemed atypical for a true respiratory gill that would normally process deoxygenated blood, right? Gills add oxygen. They don't usually receive already oxygenated blood. Furthermore, it's innervation by the facial nerve, which typically supplies the hyoid arch, rather than the trigeminal nerve, which usually innervates mandibular arch structures. That also seemed to argue against a mandibular arch origin. This led to the mandibular confinement hypothesis, suggesting that the jaw evolved as a distinct gill-less oral apparatus, separate from the gill system, and any similarities to gills were maybe secondary adaptations.
Okay, so adult anatomy seemed to contradict the leftover gill idea.
Exactly. This created a significant puzzle. Was the pseudobranch a true remnant of an ancestral gill-bearing jaw, or a secondarily specialized structure from a different arch with a different primary function? The answer has profound implications for how we understand the very origin of jaws.
So this was a genuine scientific puzzle, with different lines of evidence pointing in seemingly conflicting directions. How did a very recent study, specifically on skate embryos, Leucoragia arenacea, help to resolve this long-standing debate? And in doing so, bring us closer to understanding the ancestral truth about our jaws?
Right. This is where cutting-edge developmental biology comes into play, providing what the researchers termed unequivocal evidence. The study by Hirschberger and Gillis in 2022 used a suite of advanced techniques to pinpoint the pseudobranch's true origin and function. And they effectively settled the debate, reaffirming the classical view, but with modern, rigorous data.
How did they do that? What techniques did they use?
Well, first, they employed direct cell lineage tracing. This is a really powerful technique where you can label specific embryonic cells very early in development with a marker, like a fluorescent dye, and then you follow their fate as the embryo grows and the structures form. It's like putting a tiny GPS tracker on cells to see where they end up. They microinjected a lipophilic dye called CMDI into the presumptive mandibular arch of skate embryos.
Lipophilic, meaning fat-loving.
Yeah, these dyes are special because they integrate into cell membranes and are passed on to daughter cells when they divide, so you can track whole cell populations. They also analyzed the expression of a gene called FOXL2, which is expressed in the gill-forming epithelium of the pharyngeal arches. It acts as an early marker for gill tissue. Through these methods, they conclusively demonstrated that the skate pseudobranch is mandibular arch-derived.
So definitely from the jaw arch, not the one behind it.
Unequivocally. This direct embryonic observation is absolutely critical because, as they note, adult characteristics like blood supply or nerve innervation can, and often do, change during development as tissues mature and specialize. What looks atypical in an adult might have a very different configuration in an embryo. Tracing cells from their earliest stages reveals their true origin. They found CMDI-positive cells throughout the pseudobranch, including its lamellae and vasculature. This directly refutes the idea that it came from the hyoid arch or that the jaw evolved without any gill association.
Okay, so origin settled. What about function? Did share functions with gills?
Yes, and this was the second key finding. The study revealed extensive shared functions between the pseudobranch and true gills, not just at a structural level, but at a cellular and genetic level too, going deep into their biological machinery. They found that both the pseudobranch and the gill lamellae contain these hypoxia-sensitive neuroepithelial cells, or NECs.
Hypoxia sensitive, meaning they detect low oxygen.
Exactly. Think of NECs as specialized sensory cells that detect low oxygen levels, which is crucial for any respiratory tissue. These NECs are identifiable because they co-express serotonin and SV2-specific molecular signatures. Both structures had them. Furthermore, they showed that both structures express a remarkably similar set of genes, including HIF2A, GATA2, GATA3, and GCM2. Think of these genes as the master switches controlling essential biological functions.
Okay, what do those specific genes do?
Well, HIF2A is a gene reflecting hypoxia sensitivity, meaning it responds strongly when oxygen levels are low, crucial for respiratory tissue, while GATA23 and GCM2 are associated with things like calcium homeostasis and stress response, and critically, they're linked to the parathyroid gland in tetrapods, a structure whose function is often performed by gills in fish, particularly in regulating calcium. So this broad conservation of pseudobranch and gill function at a fundamental genetic and cellular level strongly argues against the idea that the pseudobranch evolved primarily for distinct chemosensory functions, as the alternative hypothesis suggested. Instead, it supports a shared functional heritage consistent with the serial homology of these structures.
Serial homology, meaning they share a deep evolutionary blueprint.
Precisely, it's a shared developmental plan.
What's truly fascinating here is that they didn't just show a shared origin, but a shared developmental blueprint. How did they demonstrate this serial homology at such a fundamental level, really reinforcing that it's not just a superficial similarity, but a deep connection to our evolutionary past?
Yeah, that's precisely it. The concept of serial homology means that structures are similar because they arise from a repeated underlying developmental mechanism. Even if they end up serving slightly different functions or look a bit different in the adult, think of it like this. Your fingers and your toes are serially homologous. They're clearly distinct, right? One for gripping, one for balance. But they're built from the same fundamental blueprint, 5 digits. Same basic bone structure, just adapted for different purposes. The Hirschberger and Gillis study elegantly showed this by focusing on the sporacular cartilage, which supports the pseudobranch, and comparing its development to that of the branchial rays that support the actual gills.
So comparing the support structures.
Exactly. And they found that both the sporacular cartilage and the branchial rays develop using the same fundamental patterning mechanism. Both are patterned by a specific expressing epithelial signaling center called the gill arch epithelial ridge, or GAER.
Be quiet.
Huh. No, it stands for sonic hedgehog. It's a crucial signaling molecule in embryonic development. It acts like a beacon, telling cells how to develop and differentiate. It's incredibly important. In both cases, for the future pseudobranch and for the gills, sh is initially broadly expressed in the posterior arch epithelium. And then it resolves into this GAER-like ridge of sh expressing cells along the posterior margin of the respective arch. Cell lineage tracing combined with molecular markers confirmed that these cartilaginous elements, the sporacular cartilage and the branchial rays, develop from the mesenchyme.
The embryonic connective tissue.
Right, the mesenchyme that is directly responsive to signaling from this GAER. It's the same genetic instruction set guiding their formation, the same blueprint.
Okay, so the same signaling pathway builds both supports.
Yes, and to further solidify this, they conducted these ingenious experiments where they inhibited hedgehog signaling using a chemical called cyclopamine at different embryonic stages. Cyclopamine specifically blocks the hedgehog pathway, disrupting the signaling. When they did this, they observed parallel reductions in both the sporacular cartilage and the branchial rays, depending on the timing of the inhibition. This proves a shared role for signaling and patterning these structures. If you disrupt the blueprint in one place, you see the same kind of disruption in the other, demonstrating their deep developmental connection.
Wow, that's elegant experimental design.
It really is. So this strong evidence conclusively supports the pseudobranch as a mandibular arch-derived gill serial homologue. It means that gill-like structures, patterned by a conserved developmental mechanism, were an ancestral feature of the Gnathostom mandibular arch. Therefore, the pseudobranch we see in modern fish is, in fact, a vestige of this ancient gill arch-like condition, a surviving echo of a gill-bearing jaw. This directly challenges that mandibular confinement hypothesis, which argued for a distinct gillless origin of the jaw, and instead reinforces the idea of deep evolutionary conservation, that a fundamental gill patterning blueprint was present in the earliest jawed vertebrates, maybe even their jawless ancestors, and it has been retained, albeit in a modified form, in the jaw region.
So the adult oddities like the blood supply and nerves.
They're now understood as secondary modifications during development, not indicators of a different embryonic origin. It's a wonderful example of how new techniques can resolve old debates by digging deeper into the fundamental processes of life.
Part 4. Filling Romer's Gap, Hidden Fauna in Ancient Coprolites. If we connect this to the bigger picture, our journey has shown how a single ancient fish fossil, Norsel-Aspis, and a precise modern developmental study on skate embryos can profoundly shift our understanding of early vertebrate evolution. These discoveries fill in critical details in what were once vast knowledge gaps. Now let's turn to another significant data gap in the fossil record, famously known as Romer's Gap. What exactly is this gap, and why is it so important for understanding the momentous fish tetrapod transition, that incredible evolutionary leap from water to land?
Romer's Gap, yes. It's a particularly puzzling and persistent blank space in the fossil record. It refers to a period of about 20 million years, spanning the Devonian Carboniferous boundary, where there's a significant paucity in the tetrapod body fossil record. Tetrapods, remember, are our four-limbed vertebrate ancestors, amphibians, reptiles, birds, mammals, basically us and our land-dwelling relatives. During this critical period when life was making the full transition to land, from aquatic fish forms to early land-dwelling vertebrates, we seem to have very few fossils of these transitional creatures, which severely limits our understanding of their early evolution, their diversification, and crucial adaptations for terrestrial life. It's like missing a huge chunk out of a historical movie right when the most important events are unfolding.
20 million years is a long time to have so little information.
It really is. And this era was indeed a tumultuous period in Earth's history, marked by the Hindenburg crisis, a major mass extinction event. To give you a real sense of its scale, this crisis wasn't just a regional blip. It was comparable in magnitude to the famous End Cretaceous event that wiped out the non-avian dinosaurs, or even the colossal End Permian extinction, Earth's largest known mass extinction event. That one eliminated over 90% of marine species and 70% of terrestrial vertebrate species. Just devastating.
So the Hindenburg crisis was right around this Romer's Gap period.
Exactly. It didn't just cause a few species to vanish, it profoundly reshaped entire ecosystems, both on land and in the oceans. Imagine vast Archaeopteryx forests, some of the earliest complex forests, looking a bit like modern trees but with fern-like reproduction being devastated. Or the vibrant coral reefs, the oceanic nurseries of life, disappearing from the ancient seas for a long, long time, impacting countless marine species. This wasn't just a tough patch, it was a fundamental reset of global biodiversity, leaving a profound biological gap that scientists have been trying to understand. The gap in the fossil record itself is thought to be partly a sampling bias effect.
Meaning we just haven't found the fossils yet, or they didn't preserve well.
Could be both. The conditions might not have been favorable for fossil preservation during this tumultuous period, or perhaps we simply haven't looked in the right places or with the right techniques. It's not necessarily that tetrapods weren't there, it's that their remains are incredibly scarce, which is why any new find from this period is so significant.
That certainly sounds like a critical period to understand. But East Greenland, specifically the Celsius-Bjerg group, seems to offer a unique and increasingly important opportunity to peek into this elusive gap. What makes this area so special for paleontologists trying to understand this transition from water to land?
East Greenland is indeed world-renowned for its Devonian deposits because it has yielded incredibly crucial early tetrapods like Ichthyostega and Acanthostega. These specimens are among the best understood of all Devonian forms due to their completeness and sheer number, over 500 specimens collected over decades of expeditions. They provide a foundational understanding of early tetrapod anatomy. However, despite intense study for over a century, limited attention had been paid to the upper two formations of the Celsius-Bjerg group, the Stensio-Bjerg Formation, SBF, and the Obrucho-Bjerg Formation, OBF. These two formations are particularly significant because they constitute what is probably the only known truly terrestrial sequence to span the DC boundary in terms of ancient location in central Russia, a supercontinent of that time.
So land environments, not marine ones, right across the extinction boundary.
Precisely. Unlike most sequences studied for the Hindenburg crisis, which are marine, these represent ancient land and freshwater environments. Recent expeditions to Greenland from 2009-2016 specifically focused on these upper formations, understanding their unique potential. The SBF contains the youngest Devonian tetrapod fossils, making them contemporary with Russia's Tularpatan, another critical early tetrapod known for its relatively advanced limb structure. And immediately above the DC boundary, the OBF represents a deep, permanent lake deposit, making it the earliest carboniferous fossil assemblage to date known to contain complex terrestrial and freshwater ecosystems. This makes East Greenland an unparalleled window into the immediate aftermath of the Hindenburg extinction and the faunal recovery that followed. It provides a rare glimpse into the early stages of terrestrial life after a major global upheaval.
So, just like with Norsel-Aspis, they brought in that cutting-edge imaging technology we discussed, synchrotron microtomography. What did new examinations of the SBF material using this powerful X-ray scanning reveal about early tetrapods in this crucial time period?
Exactly. The analysis of new SBF tetrapod body fossil material, specifically a lower jaw ramus, a shoulder girdle, and a suspensorium, part of the jaw suspension, using PPC S-ray CT yielded exceptionally detailed data. These specimens are not only incredibly preserved, showing fine internal structures, but they possess unique characters which are unique and therefore cannot be attributed to any of the already established Greenland taxa.
Meaning new species.
It strongly suggests they likely represent more than one new tetrapod taxa, significantly increasing the known diversity for this period. East Greenland, it turns out, is home to the greatest known diversity of Devonian tetrapods globally, much more than we previously understood.
Wow. Can you give some examples of these unique features?
Sure. For example, the shoulder girdle found in the SBF material had a short clathrum and large, robust scapulocoracoid. The clathrum is a dermal bone, part of the shoulder girdle in fish and early tetrapods. A short clathrum is a derived characteristic of more advanced feature. It's shared with later, more terrestrial tetrapods like Tularpatan, but not present in older, more aquatic ones like Ichthyostega and Acanthostega. This suggests a progressive evolutionary trend towards a more robust and mobile shoulder, essential for supporting weight on land.
Okay, so more adapted for land movement.
Yes, and even more strikingly, it featured a large and flared coracoid plate with a post-glenoid extension. The coracoid plate is part of the shoulder girdle, and the glenoid is the socket where the humerus, the upper arm bone, connects. This specific configuration, with its large extension, is an unusually derived characteristic seen in the likes of Dimetrodon.
Dimetrodon, the sail-backed creature from the Permian, much later, right?
Much later, yes. Dimetrodon was an early Permian non-mammalian synapsid, a highly capable terrestrial animal known for its powerful limbs and robust shoulder musculature. Finding this feature in the SBF tetrapods implies they likely had competent terrestrial locomotor capabilities. It indicates a significant adaptation for moving on land much earlier than previously confirmed. The contact between the clathrum and scapulocoracoid was also unusual, a sort of socket-like connection not observed in any other tetrapod, suggesting a unique biomechanical solution for shoulder stability. The jaw also showed unique features, slender and curved like a Acanthostega, but with a very unique anterior margin of the syringular bone. And intriguingly, the jaw and suspensorium were more crownward, meaning more derived or closer to modern forms than the shoulder girdle was.
Ah, that mosaic evolution again. Different parts evolving at different speeds.
Exactly. It demonstrates that even within these early land dwellers, evolution was a mosaic process. This significantly fills out our understanding of the diversity of tetrapods already existing at this critical transitional time, showing a much richer and more complex evolutionary landscape than a simple linear progression.
So we're seeing new tetrapods emerging from this gap, hinting at greater diversity than we imagined. But what about the broader ecosystem immediately after the Hangenberg extinction? This is where I hear coprolites, or fossilized poop, played a truly enormous and unexpected role in solving this puzzle. How did ancient feces become a key to understanding a cryptic fauna?
Oh, they were absolutely instrumental, and it's a brilliant example of how scientists can extract incredible information from seemingly unglamorous sources. Coprolites, as you said, are fossilized species. While often overlooked by paleontologists in the past, they are increasingly recognized as miniature Konservat Lagerstätte.
Servet Lagerstätte, German term.
Yeah, it's a geological term for deposits with exceptional preservation, often including soft tissues. Coprolites rapidly liquefy, they turn to stone very quickly, often induced by bacteria and high phosphate content in carnivore droppings. This results in minimal compression and incredibly high preservation potential of their internal contents. We're talking about the potential to preserve even delicate structures like hair, scales, teeth, bone fragments, or even parasite eggs.
Wow, tiny time capsules of diet.
Exactly. This makes them incredibly valuable, providing a direct snapshot of an animal's diet. Unlike skeletal remains, which might only tell you what an animal looked like, coprolites offer direct evidence of an animal foraging in an environment, providing unique insights into trophic dynamics within an ecosystem, essentially, who ate whom and how the food web was structured.
Okay, so where did they find these informative coprolites?
Well, the OBF, the earliest Carboniferous deposit in East Greenland, yielded a large assemblage of coprolites. Over 200 specimens collected from thick coprolite-bearing beds. This was a goldmine for understanding the post-extinction ecosystem. Initially, body fossils from the OBF itself had suggested a pretty low-diversity fish fauna of 3 taxa. A ray-fin fish called Cuniognathus gardneri, an acanthodian resembling Acanthodes lipatini, and a single chondrichthyon, a cartilaginous fish. This painted a picture of a rather sparse ecosystem recovering from the mass extinction, suggesting a slow rebound.
Only 3 types of fish found as body fossils.
Right, but traditional external observations of coprolite shape alone proved to be highly misleading and inadequate, especially because many were obscured by the host rock, making it hard to discern their internal structure or contents.
So back to the synchrotron.
You guessed it. This is where PPC-SRCT again proved its worth. The team used the synchrotron to create virtual thin sections of these coprolites. This non-destructive technique, as we discussed, allowed them to peer inside each specimen without destroying them, providing detailed, high-resolution 3D internal images. The results revealed a wealth of data, including levels of deformation, internal structuring, whether they were spiral or non-spiral, and critically clear visualization of inclusions. The undigested food remains.
And this internal view changed the picture.
Dramatically. This internal observation led to the identification of 8 distinct morphotype groups for the coprolites. 8, which is significantly more than the 3 fish taxa identified from body fossils alone. This immediately pointed to a cryptic fauna not captured by the body fossil record.
Cryptic fauna, hidden animals.
Exactly. It meant there were animals in this ancient lake whose skeletons we haven't found, but whose presence was undeniable through their fossilized droppings. It's like finding a shopping list that tells you about people who lived in a house, even if you never find their remains.
That's brilliant. Did any specific coprolite type stand out?
Yes, crucially, the discovery of large and non-spiral in structure coprolites, classified as morphotype A, was a key breakthrough. Why is non-spiral so important? Well, most fish, including the ancient ones, possess a spiral gut valve. It's a corkscrew-like structure in their intestines that helps increase the surface area for nutrient absorption, and it results in feces with a characteristic spiral shape. However, tetrapods are probably the only vertebrates to lack a spiral gut valve during this period.
Ah, so non-spiral poop likely means tetrapod poop.
That's the inference, yes. Their feces would be non-spiral in shape, unlike most fish of the time. Therefore, the presence of these non-spiral coprolites infers the presence of tetrapods in this post-extinction ecosystem, even though their body fossils haven't been found there yet. This provides further evidence that Roemer's gap is likely a sampling bias effect. The animals were there, making their mark on the ecosystem, but the fossil record simply wasn't capturing them in typical ways like skeletal remains. Another morphotype, Morphotype B, was large and spiral, postulated to be from the chondrichthyon recorded in the lake, providing direct evidence of its gut structure. This really shows how much hidden information can be locked away in these often overlooked fossil treasures.
This is absolutely amazing. It's like prehistoric CSI, piecing together an entire ecosystem from its waste products. What did analyzing the actual inclusion of the remnants of meals within these coprolites tell us about who ate whom in this ancient lake? And how did it further refine our understanding of this post-extinction ecosystem?
The analysis of the inclusions was, in many ways, the most exciting and revealing part. The inclusions were exceptionally well preserved, often better preserved than the actual body fossils from the deposit itself, likely due to that rapid early lithification of coprolites, which encased and protected their contents. This allowed researchers to identify them accurately, often down to the genus level, and the data completely challenged our initial perception that the lake hosted a low-diversity recovery fauna. Instead, it revealed a stable and productive ecosystem dominated by active predation with a complex, dynamic food web.
So much more lively than the body fossils suggested.
Much more. The study clearly showed that tetrapods and chondrichthyans were the apex predators of the lake. Their coprolites consistently contained remains from multiple prey taxa, indicating a diverse and active diet, suggesting they weren't struggling to find food. For instance, the tetrapod coprolites primarily contain remains of the actinopterygian Cuniognothus, that ray fin fish, with occasional acanthodians, those primitive spiny fin fish. This tells us what was at the top of the food chain. The chondrichthyan, on the other hand, appeared to feed primarily on acanthodes, another type of acanthodian. The smaller coprolite data showed two distinct feeding strategies among the other unidentified predators. Some animals avoided large elements like spines from acanthodes, indicating they preferred softer prey or smaller fish, while others seemed to target the fin regions of Cuniognathus, possibly its tail fin, suggesting a specialized attack strategy.
Okay, so a complex food web. But did they find anything completely new in the poop?
They did, and this was astonishing. One particular large coprolite, specimen PMU 3137 This single piece was a veritable treasure trove. It contained large cranial elements belonging to an actinopterygian that was not Cuniognathus. Its teeth morphology indicated it was a predatory fish, and its size suggested a larger species, entirely new to the known body fossil record of the lake. But even more incredibly, this same coprolite contained two small lower jaw fragments, specifically a premaxilla and an ectopterygoid, which were attributable to a small tetrapod.
A small tetrapod jaw inside another animal's poop.
Exactly. These elements lacked the ganoine ornamentation typical of fish and had a single row of progressively larger tooth sockets. This was concrete evidence of two new taxa, a new large predatory fish and a small tetrapod previously not represented in the body fossil record. It's like finding two completely new species just by analyzing stomach contents, essentially.
That is unbelievable. Two new animals revealed by a single coprolite.
It profoundly shifts our understanding. It means that immediately after the Hangenberg crisis, East Greenland witnessed a rapid recovery of a terrestrial ecosystem, not a low-diversity one as initially suggested by body fossils. The coprolite data alone indicated at least 8 distinct taxa, including 2 distinct tetrapod taxa, a far richer picture. This complexity also suggests that the lake was part of an open system, possibly with a marine link, allowing for a richer diversity than a closed, isolated lake might support. The analysis of coprolites, traditionally underestimated, proved to be an unparalleled tool for reconstructing ancient food webs and discovering hidden biodiversity, literally filling in crucial missing pieces of the evolutionary puzzle that standard fossil hunting simply couldn't find. It's a true testament to interdisciplinary science, bringing these different techniques together.
Wow, so what does this all mean for us, the curious learners trying to make sense of life's incredible journey? From a googly-eyed, jawless fish with a powerful, shark-like heart, to the intricate developmental biology of a modern skate embryo. And then to the surprising revelations hidden within fossilized ancient poop, it's abundantly clear that our understanding of life's evolutionary journey is constantly being refined, challenged, and enriched.
It really is. What's fascinating here is how these seemingly disparate areas of research paleontology, with its painstaking fossil recovery and analysis, developmental biology, peering into the earliest stages of life to understand how bodies are built, and advanced imaging techniques like synchrotron microtomography, which act as a kind of X-ray vision into the past, how they all converge. They converge to tell a richer, more nuanced story of evolution. They powerfully highlight that evolution wasn't a simple linear progression, marching predictably from one form to the next, but rather a complex, often mosaic journey. Traits developed for one purpose, like evading predators, might unexpectedly pre-adapt a creature for an entirely entirely different function, like active hunting. And sometimes long-lost features or ancient developmental blueprints resurface, challenging our assumptions about what's truly primitive versus what's derived. It's a constant dance between old and new.
It really reminds us that scientific discovery is an ongoing, dynamic process. The data gaps of today, like Romer's gap or the missing pieces of Norsel-Aspis' anatomy, are truly the potential breakthroughs of tomorrow. Waiting for the right tool, the right fossil, or maybe just the right perspective. It's about persistence and, crucially, about looking at old problems with new eyes and new technologies.
Absolutely. As Tetsuro Miyashita said about the evolution of jaws, it is imperative we understand the context. With every new fossil that's painstakingly uncovered and every technological leap that allows us to see deeper, we're not just finding new pieces to a puzzle. We're fundamentally rethinking the entire puzzle itself, revising its shape, its complexity, and its astonishing pathways. It's a humbling and endlessly exciting endeavor, really.
So for you, our curious listener, the next time you think about evolution, maybe consider this. How many other simple evolutionary stories that you might have learned might be waiting to be enriched or even completely overturned? Overturned by a deeper dive into the unexpected context that new science reveals. Keep exploring, keep questioning, and you might just find a shark's heart in the most surprising places reshaping your view of the ancient world.